All right. Good evening, everybody. Welcome to the 798th monthly meeting. Very rapidly approaching 800. Excited for that. The couple housekeeping items before we start. Please check your email and look out for the registration for two upcoming EPAASM events. The November Clinical Symposium, which will be held on November 11th, and then also the Philadelphia Infection and Immunity Forum, which will be held in December. This evening, we're really lucky to have Dr. Matthew Ziegler join us for our monthly meeting. Dr. Ziegler is an assistant professor of medicine in the Division of Infectious Diseases, Department of Medicine, and also assistant professor of epidemiology in the Department of Biostatistics, Epidemiology, and Informatics. He's a senior scholar at the Center for Clinical Epidemiology and Biostatistics at the University of Pennsylvania and the Associate Medical Director of Healthcare Epidemiology, Infection Prevention, and Control at the Hospital of the University of Pennsylvania. His clinical research program focuses on the prevention of healthcare-associated infections in immunocompromised patients and the epidemiology of multi-drug resistant organisms within the hospital-built environment. His research is supported by the NIAID and the Centers for Disease Prevention and Control Prevention Epicenters program. His presentation will focus on the role of surfaces in the healthcare environment, specifically how bacteria are shared between patients and surfaces and how improved environmental cleaning may prevent healthcare-associated infections. So please join me in welcoming Dr. Matt Ziegler.
All right, thank you, Kyle, for the introduction. Let's see if I can figure out the IT here. All right, I have no financial disclosures, but one disclosure is that I'm going to present some data today that is in a preliminary stage of analysis, so it may ultimately change when the papers come out. All right, so I wanted to give you a frame of reference for this talk. So when I talk about the environment, this is the environment that I'm thinking of. I'm thinking of the healthcare environment, which is the patient's room. And up at the top, we see a patient's bed, and we can presume that maybe this is an intensive care unit. I mean, it looks a lot cleaner than many of the intensive care units in many of the hospitals, at least in our city. But those rooms are cleaned, and then subsequently a patient enters into that room, and the patient touches surfaces, for example, the bed, the bedside railing, and they're interacting by touch, but they're also potentially have body fluids that may contaminate the environment that the surface is within their room. But at the same time, there are healthcare workers who are coming into that room. They are interacting with surfaces, they are interacting with the patient, potentially performing invasive procedures on the patient in that context of the patient's room. And at the end of that patient's stay, it's possible that the surfaces within that room have been contaminated with bacteria or other pathogens. And then we have the dirty room at the bottom left-hand corner. That room then gets cleaned, and then this cycle continues. And this cycle continues for the life of the hospital. And there's an opportunity for the potential for bacteria to persist on surfaces despite cleaning of these rooms. And there may be a potential for those bacteria or other pathogens to then infect patients who come next into that room. So this is the frame of reference that I want to kind of bring your attention to is really the hospital room, the patient's room. So why should we study surfaces in inpatient rooms? What we know from prior literature is that we can readily recover pathogens from hospital surfaces, and including multi-drug resistant organisms from hospital surfaces. We also know from prior studies that cleaning of surfaces after a patient leaves a room, it's often inadequate. Some studies have reported that by gross visual assessment, up to 50% of surfaces are inadequately cleaned upon discharge of a patient from a room. And there's also some literature that supports that the patient who comes before you may impact your risk of infection. So Dan Freeberg's group has made associations with C. difficile. If the patient before you had C. difficile, you may be more likely to have C. difficile infection yourself if you're the following occupant of that room. And similarly, the amount of antibiotics that the patient receives before you enter that room may predict a risk of C. difficile infection, really suggesting that bacterial that persists on surfaces may be putting patients at risk. But what we don't know is the incidence of surface-based transmission of pathogens, particularly for multi-drug resistant bacteria. And we don't know what predicts these infections at the patient level, the patient that is actually in that room and at risk of infection. And we are also, we have room to improve as far as finding methods to improve cleaning of surfaces to prevent potential infection of patients. So for this talk, I'm going to present two different research studies. One is predominantly focused on surface contamination with multi-drug resistant bacteria and potential acquisition from surfaces. And the other is going to be focused on how we can improve cleaning to prevent infection. So the first study is dubbed EPAASM, or Infection Control, Environment, Microbiome, and MDRO Transmission Study. And this study had two aims. The first was to identify patient and environmental risk factors for surface contamination with multi-drug resistant organisms. So what predicts contamination with MDROs? The second aim is really focused on the microbiome of the patient and the microbiome of the room. So how do the microbiomes interact with each other over time? And to identify factors that kind of contribute to the sharing of bacterial taxa between patient anatomic sites and environmental sites. So split into culture and microbiome aims for this study. To conduct this study, we enrolled patients at the Hospital of the University of Pennsylvania, HUP, and at Penn Presbyterian. We enrolled ICU patients from both medical intensive care units and surgical intensive care units. And our goal was to try to capture patients really shortly after they enter into a new room, a new intensive care unit room. So that is kind of the frame of reference is a new potentially clean room. We then sampled patients and their environment longitudinally over their stay. And we wanted to enrich the population for patients who are at risk for colonization with multi-drug resistant bacteria for this study. So we only enrolled patients who had a history of a prior MDRO culture in the preceding 30 days. This study started in November of 2019. We took a brief break around March of 2020. And then we eventually finished the study in April of 2021. And we enrolled a total of 102 subjects with 278 sampling events. And as I mentioned, we sampled both patient anatomic sites and environmental sites within their room longitudinally. And we had dense sampling in the first week of occupancy of that room, and then continued weekly until day 28 or discharge. So once a patient left the room, we stopped sampling that patient in that room. The sampling strategy that we used was to sample environmental surfaces in 3 composite groups. And the rationale behind these groups was a near patient group of sites. And these really reflect the types of surfaces that a patient may touch themselves in that kind of space that is within arm's reach. So that's the bed rail, the call box, the remote, the overbed tray table. So that grouping of surfaces. The distant group of surfaces are really surfaces that are more likely to be interacted with by healthcare personnel. The IV pump, the med card, the keyboard and the mouse of the in-room computer. And then the final group of environmental surfaces included bathroom surfaces and wastewater surfaces, toilets, sinks, and other kind of hard surfaces in the bathroom environment. And then for patients, we sampled multiple anatomic sites for each collection episode. So all of these sites were sampled with each collection episode. We sampled the nares, the oropharynx, the inguinal area, perirectal swab, and stool. And the intent in getting so many anatomic sites was really to increase our sensitivity to detect colonization with multi-drug resistant bacteria or potential acquisition of multi-drug resistant bacteria for patients occupying those rooms. And the processing of those specimens really were kind of parallel processing for the two different aims, the culture-based aim and then the microbiome aim. So for the culture-based aim, each of those cultures, composite cultures or anatomic cultures, underwent culture using selective media to recover methicillin-resistant Staph aureus, vancomycin-resistant enterococci, carbapenem-resistant Enterobacterales, extended-spectrum beta-lactam-producing Enterobacterales, and C. difficile. So we use multiple selective media to collect a variety of multi-drug-resistant bacteria from both environmental and patient sites. Those bacteria that were recovered from culture were identified with MALDI-TOF and then eventually made it to our freezer. From there, they were recovered and underwent whole genome sequencing so that we could verify the relatedness of environmental and anatomic specimens, bacterial culture specimens. And then the parallel process on top is that we performed 16S sequencing of these specimens as well to characterize the microbiome of the anatomic and microbiome and the environmental sites. And I'm going to share with you the first attempted analysis that we performed of this data. And for this, I'm going to start by just sharing with you the environment. So for a moment, we're going to ignore the patient and focus on the environment that surfaces within the intensive care unit. And what we did for this analysis was we were interested in the potential characterization of the room by distance from the patient. So the patient is sitting in the middle of the room, and the head of their bed is really what we considered kind of the epicenter of their risk of contamination within the room. And we measured distance from the patient's head of the bed and how far away we got from that patient's head of the bed to each sampling site. And as we got farther from the patient's head of the bed, we got closer and closer to wastewater sites, the bathroom sites. So we designed a measure of distance from the patient to where we sampled in the environment so we could characterize the bacteria and the microbiome at each of those sites as it related to distance from the patient. And the outcome of this analysis was both culture-based detection of multi-drug resistant bacteria and bacterial DNA deposition by characterizing the microbiome of these specimens. And what we found was interesting. Here I'm going to highlight first the gram-negative organisms that we recovered by culture. And again, these are all of the bacteria recovered by culture are drug-resistant bacteria. And what we see is that as distance increases from the patient's head of the bed, as we get farther from the patient and closer to the bathroom, we have increased probability of recovery of these multi-drug resistant gram-negatives. So you can see, for example, Acinetobacter, Citrobacter, Enterobacter, Pseudomonas, Klebsiella, Stenotrophomonas, all of these are increasing in probability of detection as we get further and further from the patient and closer to the bathroom sites. But in contrast to the gram-negatives, we see for gram-positives the opposite association, where we see a higher probability of recovering these bacteria closer to the patient and a decreasing probability of detection as we get closer to the bathroom sites and farther from the patient. And this is aggregated here in a more dramatic fashion, that gram-negatives, as I mentioned, are increasing in probability as we get to the bathroom sites, and gram-positive decreasing as we get to the bathroom sites. Now, in contrast to culture-based data, this is recovery of bacterial DNA. And again, this is now the microbiome of the environmental sites. So this is now no longer selective for multi-drug resistant bacteria. These are aggregated at the level of the genus. And we see some differences here, where some gram-negative taxa are really persistently distributed throughout the room. For example, if you look at Pseudomonas, we no longer see that association where we see increasing probability of detection as you get close to the bathroom. We see it deposited everywhere. So Pseudomonas DNA deposited at all of the environmental sites. And similarly with Acinetobacter, we see very high probability of detection of multiple sites, close to patient, distant, and farther from the patient. However, there are some bacteria where that association is still matching what we found with culture. For example, with Clostridium, we see a slight decrease in detection as we get further and further from the patient. And similarly with Staphylococcus and Enterococcus. So what did we find? What is the summary of this, first analysis of the data? We found that multi-drug resistant organism detection varied by distance from patients within ICU rooms. The probability of detecting gram negatives increased as we got farther from the patient and closer to the bathroom, while in opposite, C. diff and MRSA were closer and nearer to the patient sites. This discrepancy of culture and DNA-based detection we found interesting, particularly that Pseudomonas and staph DNA was distributed everywhere in the hospital room, but there were really these limited areas where we were able to culture multi-drug resistant bacteria, methicillin-resistant Staph aureus, carbapenem-resistant Pseudomonas. So perhaps what this is showing us is that there's deposition of these bacteria, but the ability to recover by culture suggests that at those particular sites, cleaning measures that are taken may be inadequate at those particular sites, or there may be features of those particular environments, particularly the wet environments of the bathroom, that may allow for persistence of these particular bacteria at those sites. So that is, that discrepancy we found interesting. So that was just limited to the room. What if we introduce the patient to the equation and we compare the patient and the room data that we recovered from the study? So this is just a 1,000-foot view of the specimens that are included from the study. This is a principal coordinate analysis of Brake-Curtis dissimilarity, a measure of beta diversity of the specimens. And the anatomic specimens are all red, and the environmental specimens are all greenish blue. Perhaps I'm colorblind, and that may look different to you. And what you can see, if you were perhaps seated a little bit closer to the screen, is that there is a large cloud of red specimens that are kind of relatively well circumscribed as the anatomic specimens. But there are some of these blue specimens that are falling well centralized within that cloud of anatomic specimens, suggesting that there are environmental specimens that are looking very similar to anatomic specimens. And similarly, when we look at the cloud of environmental specimens, we see red dots also distributed within that. So there are some anatomic specimens that are looking very similar to environmental specimens. So this is giving us kind of just a very quick first view of how the microbiomes are looking and comparing from anatomic and environmental specimens. But we can break this down into multiple levels and explore this further. And this here is just broken down by the different sites that we swabbed in comparison. For example, how does the bathroom site compare to the inguinal site of the patient? Or how does the nearest patient site compare to the nasal site of the patient? And what I think is interesting to highlight here is not necessarily the averages for this graph, but the outliers. And you can see in this case, the y-axis is Brake-Curtis similarity. So how similar are the microbiomes? And for some of these specimens, if you look at the near site and the nasal site, there's almost 60% similarity in that particular outlier between an anatomic and an environmental site. So while on average the similarity is lower, there's certainly a range here of similarity and dissimilarity of the microbiomes of these specimens. We were interested in delving into this and figuring out what predicts similarity or dissimilarity. What can predict why a microbiome of a patient in an environmental site may be similar? And the first thing that we looked at to investigate this particular question was time. Does time, the amount of time that a patient is residing in a room, does that predict the similarity of that patient from their anatomic sites to their environmental sites? Does that make them more similar or less similar? And what we see here is that with increased time of occupancy of the room on the x-axis, during the first week of their occupancy of the room, we're seeing that the microbiomes of the anatomic and environmental sites are becoming more similar. And this is broken down into the different environmental sites. And what we can also see is that the similarity differs by your distance from the patient. Near patient on the left here, we see that the y-intercept is higher, suggesting that at baseline, even at the day 0, day 2 specimens, the microbiome is already more similar at the near sites than to the bathroom sites, which is on the far side, the right side of this graph. But in all, we see an upward trend to these lines, suggesting that there is increasing similarity of the microbiomes over time. And one thing that's important to note here is over time, these rooms are subject to daily cleaning. So each day there is an environmental service staff who comes into the room and wipes down surfaces with disinfectant. And despite daily cleaning, we're still seeing this trend of increasing similarity over time. So in summary, the patients in the room microbiomes are becoming more similar over time. That varies by distance within the room. And we were also interested in investigating, well, as similarity increases in microbiomes, does this predict at all the similarity by culture? Is microbiome a good measure of what we're seeing with colonization of multi-drug resistance organisms in patients and detection from environmental sites? And what we found was that a 10% increase in Bray-Curtis similarity, so 10% increase sharing of taxa between environmental and anatomic sites was associated with a roughly 3.5% increased probability of finding the same multi-drug resistant organism in environmental and anatomic sites. And we tried to get even more specific with this comparison by using whole genome sequencing data to link the matching bacterial cultures. And what we see there is that the probability is slightly lower, but still with a 10% increase in similarity of the microbiomes, a 2.5% increased probability of detecting the same multi-drug resistant organism using whole genome sequencing data. So the next question that we had from this data was, we see that the similarity is increasing over time. Are there clinical features or cleaning features that can predict the sharing of microbes between surfaces and anatomic sites? For example, cleaning. I mentioned to you that rooms are cleaned daily while patients occupy the rooms, but also there's a difference in the type of cleaning that is performed prior to entry into the room. One of the things that is employed is ultraviolet germicidal irradiation, or UV light, in rooms prior to patient occupancy. And we were interested to see if that could impact the microbiome of the surfaces. Similarly, could there be features of the patient care that could influence the sharing of microbes between surfaces and the patient? And specifically, this is part of the analysis that is ongoing. I'm going to share with you a limited number of variables that we've investigated, and that includes the potential for certain types of catheters to potentially prevent contamination of the healthcare space. So I'm a healthcare epidemiologist, and we think of catheters causing infections, but there's also the potential that a rectal tube, a fecal management tube that contains the feces of a patient, may potentially decrease the contamination of the environmental space surrounding that patient. Similarly, a urinary catheter contains urine output from a patient and then thus may decrease the contamination of surfaces surrounding that patient. Sedation would mean that a patient is less likely to get up out of their bed and touch surfaces that may be more distant from their room. So these are some of the things that we were looking at as kind of a first pass analysis. Sorry. In addition to that, we were interested in the impact of antibiotics as well. I'm going to share with you two tables here. The first is looking at these clinical predictors and how they impact the similarity of microbiomes. And then the next slide, I'm going to share with you how it shared the similarity by sharing of culture. Looking at these clinical predictors with an outcome of Bray-Curtis similarity, how similar are the microbiomes? First, I looked at UV cleaning prior to entry, and this did not significantly impact the similarity of microbiomes between patient and environmental sites. However, when we looked at these invasive catheters that patients receive that may contain body fluids, we see on average that these are decreasing the similarity between the patient environment and the anatomic sites. With the fecal catheter really having the largest estimate of a decreased, patients with a fecal catheter have a decreased similarity of roughly 2%. And patients who are sedated have a decreased similarity of roughly 3%. those patients who are sedated compared to patients who are not sedated. And one other variable that I have here is patients who are eating. So patients who are eating, have an oral diet, are more similar to their environment. There are likely some other confounders that go along with this. Patients who are eating are likely to be more well, maybe moving about the room, but they are also less likely to have a fecal catheter. So they are eating food and likely having bowel movements within this room. That could also be a predictor for why we're seeing, an explanation for why we're seeing oral diet as a predictor of similarity between these sites. Whereas that table was looking at microbiome similarity, this table is looking at sharing the same multi-drug resistant organism at an anatomic and an environmental site. And what jumps out here at the top is that rooms that underwent UV cleaning prior to patient entry are much less likely to have a shared multi-drug resistant organism from patient environment and anatomic site. We'll delve into this in a little bit more detail in a second. But UV cleaning prior to entry was, it kind of jumps out here at the top. We see a similar trend to the estimates, but not meeting the same level of statistical significance for these catheters that contain body fluids. But we do see a trend where urinary catheters, for example, decrease the odds of having a shared multi-drug resistant organism from environmental and anatomic sites. But on average, here we see that we have less power to detect a clinically significant impact of these particular predictors. And I mentioned that I was going to get into the UV cleaning in a little bit more detail. When I think of the potential impact of UV on a patient's room, what I envision is that the UV should decrease the potential viable bacteria on surfaces really on that first episode that the patient enters into the room. Subsequent to entering that patient's room, then we have both the impact of the potential prior room occupant, residual contamination on surfaces, and then new contamination from that patient or potentially from other patients in the surrounding area where healthcare workers may be entering in and touching surfaces and potentially contaminating surfaces. So for this analysis, I really just focused in on that first collection episode within 24 hours of entry into the patient's room. And what we see is that when we look at UV cleaned rooms, 40% of those rooms had a positive multi-drug resistant organism environmental culture, compared to 57% where UV cleaning was not utilized. That still could be the result of patients contaminating those surfaces in that very brief period of time that the patients have entered the room. So again, this is after the patient has entered. So to investigate this a little bit further, what I did for this analysis was I removed any matching culture that both the patient had and the environmental surface had on that first collection episode. So if that patient contaminated that room in the first, say, 4 hours of entry into that room, we should find that multi-drug resistant organism at one of these multiple anatomic sites that we found. If we found it in the environment, but we didn't find it in any anatomic site, that may suggest that that's residual contamination from the prior room occupant in that particular healthcare space. And what we see is that if a room was UV'd, we found residual, potential residual contamination in 18% of rooms, compared to 36% in rooms where they did not undergo UV cleaning. So in this particular analysis, we see that UV cleaning prior to entry is associated with a significant odds ratio of 0.39 of detecting this potential residual multi-drug resistant organism on surfaces. When we look not at culture, but we look at microbiome data, we look at bacterial DNA on surfaces, this association then breaks down. We no longer see an impact of UV cleaning on unique ASVs or amplicon sequence variants or aggregated read counts at those sites. And this may be somewhat similar to what we saw before when we were just looking at the difference between culture and microbiome of environmental sites, is that there may be deposition of bacteria on these surfaces, but there may be disinfection of the surfaces and thus not recovery by culture. So that is where this kind of nuance, the difference between bacterial culture and microbiome detection may give us some hints as to where there are opportunities to improve cleaning. So the next step and the next question that we had asked with this data is, why are the microbiomes becoming more similar over time? Could this be because of an independent change in the microbiome of, say, just the anatomic specimens, where the environmental specimens are staying the same? Or could there be sharing of both bacteria by culture and sharing of taxa by looking at the microbiome between anatomic and environmental sites? So to do this from a microbiome standpoint, we looked at the transfer of ASVs, or amplicon sequence variants, and we defined two different transfer events. Environment to anatomic transfer means that we first detected that ASV in the environment and not at any anatomic site, but then at a subsequent period of time, we detected that at an anatomic site. The flip of that is anatomic to environment transfer. We first find it at an anatomic site, not anywhere in the environment. And then on a subsequent day, we find it in the environment. And that suggests that it started on the patient and then subsequently contaminated surfaces. So this is just an example here, just to kind of spell this out, because I know this is somewhat word soup here. So this is an example of how we would define an environment to anatomic transfer. On day one, for example, we may have Pseudomonas that we recover from an environmental site, but we don't find it anywhere on the multiple anatomic sites that we've sampled. Similarly, we find that that pattern on day two, where we find it in the environment, but nowhere in anatomic sites. And then day four, we find it in both. And so that suggests that that particular Pseudomonas was in the environment and then subsequently acquired by the patient when we recover it at the anatomic sites. And so that is kind of how we are defining these transfer events between the environment and anatomic sites or the patient. So this here is really trying to quantify how often are we seeing transfers using microbiome data, looking at ASVs. This is the fraction of taxa, or defined as ASVs, transferred from anatomic sites to environment. And this is count of subject on the y-axis. So you see a large bar on the left at 0, suggesting that over 20 patients, we did not identify that they shared or spread any of their anatomic taxa to their environment. So they were the non-contaminators. But we have this distribution then to the right of patients who up to 40% of the bacterial taxa that we found on their anatomic sites contaminated their environment. So those, while we have a large count of non-contaminators, we have some heavy contaminators at the right end here of this figure. And then we can flip this to who's picking up environmental taxa. And we see a similar but a different scale here. Similar story, but different scale. Over 20 patients here on the left are not picking up anything from the environment. But we have some patients who are picking up almost 8% of the environmental microbiome into their anatomic sites. So there is a distribution of patients here who are picking things up from surfaces in these patient rooms. And we see a similar story, but with smaller quantity when we really hone into what can we recover by culture and limited just to multi-drug resistant bacteria in these rooms. So this is just count of anatomic to environment transfer or environment to anatomic transfer by culture. And you can see here, the blue lines are the shedding, the contamination of the environment, where we had 10 counts of Pseudomonas being first recovered from a patient, and then at a subsequent time point, contaminating their environment. But we also found that patients were picking up multi-drug resistant organisms from surfaces at a lower rate. We saw events where Pseudomonas was detected first in the environment and then subsequently acquired at the anatomic site, similarly with Enterococcus and Stenotrophomonas. And these are multi-drug resistant, in most cases carbapenem resistant, gram-negatives, and then vancomycin resistant Enterococcus here. So we're seeing this kind of mirrored where it is more common to detect the contamination of the environment, but we are still seeing picking up of taxa from the environment to anatomic sites. And so the next question that we had was, well, what predicts the shedding of bacteria to sites, the anatomic to environment transfer, or the picking up of bacteria from environmental sites, the environment to anatomic transfer? And this is a figure of antibiotic exposure on the y-axis, and then it's broken down into the two different types of transfer. And what we see here is that with increasing exposure to antibiotics, this is quantified as antibiotic days, so the number of antibiotic types that you would receive in any day aggregated over a 7-day period. As you are exposed to more antibiotics in that week prior to sampling, it means that you're more likely to pick up bacteria from surfaces. So the exposure to antibiotics really primes you to pick up these environmental contaminants. In contrast, the more antibiotic that you get, the more potentially that reduces the diversity of your own microbiome at anatomic sites, and then the quantity of ASVs that you're shedding to the environment is actually decreasing. So antibiotics increase the acquisition of environmental taxa, but decrease the contamination of environmental taxa when looking at the microbiome. So in summary, the patient and the surface microbiomes are increasing in similarity over the first week of occupancy. Ultraviolet germicidal irradiation prior to patient entry significantly reduces the odds of this potential residual environmental multi-drug resistant organism contamination of surfaces at that first sampling event. And we've also shown you just in this preliminary analysis that there are several clinical factors that are associated with either similarity of the patient and the environmental microbiome or transfer of taxa or ASVs from environment to patient or vice versa. Where invasive catheters make you less similar to your environmental, the microbiome of your environment, whereas the patient who is not sedated and eating is more similar to their environment, and that antibiotics can predict these environment to anatomic transfers, particularly with exposure of antibiotics priming you to acquire environmental pathogens. So that is the first kind of half of the talk, but I'm already at 38 minutes. So I'm going to just go through relatively briefly the second half of the talk. So that was the investigation into how patients are contaminating and interacting with surfaces in their room. The second half of the talk is how can we improve patient outcomes related to this risk of acquiring pathogens from surfaces? And this is the SHINE study, stopping hospital infections with EVS or environmental services. So I wanted to go back to kind of a brief explanation of how hospital rooms are cleaned in a little bit more detail. One important point to make is that cleaning is a relatively complex multi-step process to clean hospital rooms. It involves both the cleaning step, which is the physical removal of material and soil, which must be performed before disinfecting of surfaces to have a good result. So we must first remove the biofilm from surfaces, the secretions from patients, the body fluids from surfaces before we then attempt to disinfect those surfaces, which is the inactivation or killing of the pathogens or microbes on those surfaces. And really, it requires both of these to be effective. And routinely, there are two primary stages of cleaning. There is the daily cleaning that occurs after a patient is entered into a room, where the surfaces are cleaned and there's mopping that is performed and their disinfectant is applied. And then there's terminal room cleaning, which is the more rigorous cleaning that occurs after a patient leaves the room and before the next patient comes in. So that is terminal room cleaning, and that is really the emphasis of this particular research project. And when terminal room cleaning occurs, there are a few different modalities that we can use to assess how effective the cleaning was. One is to look at the surfaces after someone cleans them and say, is that dirty or is that not dirty? That's called visual monitoring, which is really have been the mainstay of cleaning assessments for a long time. The other methods that are available is the application of a UV marker. So you put a UV marker on a surface Then someone cleans the surface, and then you use a UV light to see if that marker is still present after cleaning that surface. If you still see light, it means that it wasn't cleaned adequately, and that's something like glow germ, for example. And then the next one to mention is ATP monitoring. So we're using the detection of ATP as a suggestion for biological material on surfaces. And so there are proprietary tools that can be used to measure in a quantitative fashion how much ATP is on a surface with a bioluminescence technology. And so the way that this works in practice is you swab a surface, and then it goes into a luminometer, and it can give you an output as to how much ATP is present on that surface. One of the benefits of this particular modality is that you don't need to sneak into the room before someone cleans the room to be able to assess how clean that is. So it somewhat protects you from the observer bias of, I'm going to put some UV markers all over this room, and let's see how good of a job you do when you clean the room. So that's one of the other benefits of this technology. But what we were interested in is we have these technologies, and we know that rooms are often inadequately cleaned, but we did not know if we can use these technologies to actually improve not only how clean the rooms are by culture, but actually clinical outcomes for patients. So the impact of this study was to see, does improved cleaning actually result in decreased colonization and infection with multi-drug resistant organisms in patients? So to do that with an outcome that is relatively less common, we needed a larger number of centers. And so this was a cluster randomized crossover trial that we performed at 3 medical centers here in Philadelphia at the Hospital of UPenn, Barnes-Jewish in St. Louis, Stroger at Cook County in Chicago, and enrolled medical ICUs and surgical ICUs at these centers. And the intervention was performed, both interventions were performed in a randomized order. And the intervention was to randomly select at least 5 rooms per week at that ICU, and to monitor after terminal room cleaning occurred, and to provide direct feedback to the cleaning staff, the environmental services staff, to tell them how good of a job they did with cleaning of that room. And we compared the 2 methods using ultraviolet markers, the glow germ, to this ATP technology to determine how much ATP was residual on surface after cleaning occurred. And so we are assessing the quality of cleaning, providing direct feedback to the EVS staff that cleaning was either adequate or inadequate, and then surfaces that were inadequately cleaned were recleaned until they met the marker of adequate cleaning by either modality. And not only real-time feedback, but also weekly reports of the results of either method were provided to the EVS staff and to their managers. So the hope with this intervention was that it would spread not only to the specific observations that were occurring, but by providing both structured real-time feedback and aggregate feedback, that cleaning would improve globally on the unit, even in rooms where observations were not being performed. And the surfaces that were assessed were borrowed from the CDC checklist for terminal room cleaning. And as I mentioned, the primary outcome of the study was actual clinical outcomes of the patients on these units. So it was implemented in a cluster randomized unit-based fashion. And we looked at the rate of colonization or infection with multi-drug resistant organisms and patients occupying those units where the intervention was being performed. So we were looking at MRSA, C. diff, VRE, and multi-drug resistant gram-negative bacilli. And we had relatively strict definitions of infection using infection control definitions from NHSN, or the National Healthcare Statistics, so we could actually define the National Healthcare Safety Network definitions of infection. So we could have a uniform definition of infection. And we had applied windows of time where any potential positive culture would be applied to their ICU stay. And so we looked to see if the particular intervention using a UV cleaning intervention or an ATP monitoring and feedback intervention on unit was associated with either a decrease of colonization or infection with these particular pathogens. And so this is the allocation of the interventions in a kind of a graphical picture, just to make it a little bit clearer. We have a baseline period where there's no intervention that is being performed, and this is visual monitoring. This is, does it look clean? And then at study onset, they're randomized to either start with an ATP monitoring or a UV monitoring, and then there's a washout period, and then they get the other intervention at the end. So everybody got both interventions. And what we found was that with the UV monitoring modality, there was an incidence of 7.9 infection or colonization events per 1,000 patient days, compared to the ATP intervention at 5.2. When we looked at just infection, the differences were smaller, where UV, during the UV period, MDRO infection was 3.7 per 1,000 patient days compared to 3.2 on aggregate across the multiple institutions. What we did to analyze the data was to perform a mixed effects Poisson model with random intercepts by patient location or hospital. And what we found was, similar to the summaries that I gave above, that the incidence rate ratio of MDRO infection or colonization was reduced with the ATP intervention with an incidence rate ratio of 0.87. But again, when we looked at just infection, not infection plus colonization, we did not find a statistically significant impact of the intervention on infection alone. One of the things that we looked at is kind of a potential downside to this, intervention was it may take more time. And in time and room turnover is an important metric for hospitals because they need to get patient rooms turned over so that they can care for more patients. But what we found was that that the impact on average of room turnover time was fairly minimal. So this is in minutes in this table. So on average, there was a 1 minute increase in turnover time for ATP rooms compared to baseline. And this is because the intervention is not applied at every single room. So on average, if you spend a little bit more time on one room by doing an assessment and providing feedback to EVS staff, the rooms where that is not where that is not being done, there's not an active assessment, you may still get that benefit of the improved cleaning that particular EVS staff has learned from the prior observations, but it may not take as long because you're not providing that feedback during that session. So it does increase time, but the amount of time that it increases is quite small. So in summary, the ATP intervention, so this is ATP monitoring of cleaning effectiveness with both real-time and aggregate feedback, was associated with a reduction in multi-drug resistant organism infection and colonization with these adjustments for the statistical model. So one of the limitations of the study was that we actually had a lower than anticipated event rate. We actually thought we would have more infections in the study than we did. So we were relatively underpowered for the outcome of infection alone. So we did not find any statistically significant impact of the infection. We thought there would be more infections during the study. But one of the other kind of takeaways from this study is that we've drawn a link, an association between effectiveness of cleaning and real clinical outcomes for patients. If you clean rooms better, they have fewer infections with multi-drug resistant organisms, which is really one of the takeaways of the study. So I think, Laurel, you were asking this, should you eat your lunch after it falls on the hospital floor? Probably not, but we can model it. And so now we have all of this data so we can model the probability of acquiring a multi-drug resistant organism from the sandwich that you dropped on an ICU floor. I don't think you needed this model to know the answer to that question, but this is the power of statistics. So, but in seriousness, understanding surface contamination, I believe, improves our kind of knowledge of how we can impact cleaning, and improving cleaning prevents infection for patients in our units. So we have a few kind of upcoming studies that are related to upcoming or ongoing studies that are related to the study of surfaces and patients. One, which I was starting soon, is looking at not only the room where the patient resides, but the other locations within the hospital where patients travel. So they travel to the CT scanner, to physical therapy, occupational therapy, and this network of potential contamination and transfer of bacteria, it may be larger than just the narrow view of the patient's room where they spend the majority of their time. We also have Eb Lautenbach is leading a study that I'm collaborating on, looking at patients' homes where patients are colonized with ESBLs and may transfer ESBLs to other occupants of that home. So looking at potential predictors of transmission of these resistant organisms in the home space. And also Brendan Kelly, who has been very instrumental to all of these analyses, is working on getting a project started. We're looking at the role of FMT to address colonization with multi-drug resistant organisms and to see if FMT can not only prevent persistence at anatomic spaces, but also decrease contamination of the environment with multi-drug resistant organisms. So another potential modality to decrease contamination of the environmental space. So that is the talk. So thank you to our research team. A special thanks to Brendan, who has been a great mentor for all of these analyses, and the rest of our Ares research team, and the CDC for funding this research. All right. And we've got 8 minutes for questions. Yeah. So the first question, do you have similar data for C. auris and improvement in patient colonization using ATP to improve room cleaning? So a good question that we did not investigate for this particular study. Fortunately, we do not have an abundance of C. auris at our hospital. But in the Midwest, predominantly in Chicago, they do. And so we did not investigate C. auris for this particular study. I think at the time that we did the study, that was not quite as prevalent, but that is an excellent reminder that I should bug our Chicago colleagues to make sure to see if we have data for C. auris going back into the time when we started this study. But I do not have that data. And the other was not a question, but thank you, Irv. So for this study, we did not enroll patients who are actively on precautions for COVID-19. That was mostly a limitation at the beginning of the study. We didn't know if we could process the specimens in our lab, and so it was more of a safety concern that ultimately was not really a problem. So we did not have patients with COVID-19 precautions. There was variability as to whether or not the patient was on precautions at the time, dependent on what pathogen they had. So for our infection control purposes, certain multi-drug resistant organisms result in contact precautions only for the duration of that index hospitalization, and then are not necessarily reused for a subsequent hospitalization. So for this case, there is variability of whether or not the patient themselves were on contact precautions. That is one of the variables that we plan to analyze in the data to see if patient contact precautions impact the environmental microbiome, with the primary interest in, there's 2 ways that could potentially infect the environment. Some people have suggested that if you're wearing precautions, you're more likely to do things that you wouldn't do if you weren't wearing precautions. Potentially more likely to actually have contamination of the gown and the gloves and then spread that throughout the room. And there's also the possibility that that may decrease the contamination from the healthcare worker, the contribution of the healthcare worker microbiome to surfaces. So something that we have planned to analyze, but will be somewhat limited in our ability because of the the sample size. The majority of the patients are on isolation. So a very good question that I don't have a exact answer for, but I can hypothesize kind of what I think is. I think it's more the former. Some of the surfaces that are in that distant category are the IV pole, the and the computer that the healthcare worker, predominantly the nurse, is using very frequently. But other healthcare personnel are using that in-room computer. The visitors are unlikely to interact with the computer because they shouldn't have a login, but a lot of them will interact with the IV pole as it starts to beep. So there is the potential that visitors. We did not record any data on visitors. Who else could be in the space? So it is possible that as time goes on, that there's not only healthcare workers, that kind of cloud of people that interact with surfaces grows, not just from healthcare workers, but also potentially visitors. So I would guess it's more the former, but it's something that we did not specifically investigate. I think we've got two questions here. So that is another variable that we have not yet analyzed, but is a very good question. Not only interested in the immune system of the patient, but also other medications that they might receive. For example, proton pump inhibitors or other things that might alter the microbiome of the patient and prime them for acquisition of environmental bacteria. But that is something that we plan to investigate, but have not yet. So that is part of the preliminary analysis. So I do have that data, and I cannot remember off the top of my head, but I do exactly have that data of which, of the bacteria that we find on surfaces that are not matched to anatomic specimens on that same day, what particular bacteria those are. I have that data, but I am not able to recall exactly that breakdown, but that is something that I can definitely report back on. But that also reminds me of one of the future directions of this work is actually to get into the rooms before the patient. So to follow the room longitudinally, not only for one admission, but patient stays in that room, they discharge, and then to follow the room rather than the patient. So that way we can find out who deposits bacteria to surfaces, which bacteria persist on surfaces, and for how long during that subsequent room stay. So that is a question that I very much want to investigate further. We weren't able to answer in quite so easily in this study because it's hard to get into patient rooms in that very brief gap after someone leaves and someone comes in. The healthcare system doesn't want us delaying admissions, but we will try. So I think that there's several things that we kind of I can hint at, but we don't have kind of the complete data just yet. I think there's two things that we can focus on. One is the particular microbe, and then the particular space. So one thing that has been reported on previously is that if there is a patient who might potentially contaminate a wastewater reservoir with a multi-drug resistant Pseudomonas, that Pseudomonas gets in there at some point. Usually we presume from a patient specimen that kind of contaminates a wastewater site. That may then prime that environmental reservoir to persist with that multi-drug resistant organism for a period of time that may subsequently infect patients. So if you know a patient has a multi-drug resistant Pseudomonas, maybe you clean the wastewater sites more aggressively with that index admission to prevent kind of the persistence in that site. And similarly, if you have a patient with C. diff or staph, maybe you need to clean those near surfaces a little bit better. So I think that that's my hope, is that we can figure out ways to be more efficient in our cleaning, to kind of hone in on specific areas, kind of in combination with the particular pathogen. But also that we struggle to improve the cleanliness of surfaces during the time that a patient resides in the room. The microbiome is becoming more similar, and it's hard to clean a room and to maintain kind of clean surfaces while a patient is actively occupying. There are ways potentially to address that. For example, long-acting surface disinfectants have been investigated. Patient hygiene is actually another kind of way to potentially improve that, is another thing that we can investigate. Hand hygiene for patients is kind of an area that some others are researching in the Epicenters group. So I think there's kind of two parts to future directions that we might be able to take from this research. And then I think, Brendan, you had a question. All right, I think we've got any data of between room colonizations, which is a local stationary crew. Okay. So the question is, I believe, is a local like nursing crew or a patient care crew could potentially spread bacteria from one room to another in this kind of more limited kind of neighbor to neighbor spread. This study wasn't very well primed to analyze that because of the way that we enrolled patients. We enrolled patients as they met eligibility criteria, and those were separated in space and time, different units in different times. So we didn't necessarily have pairs of, say, neighboring units that were enrolled at the same time. So we weren't necessarily able to see if an MDRO that was discovered at an environmental site that didn't belong to the patient, if that was related to the neighbor, for example, or the nurse of that particular patient. So we weren't able to answer that. That it's a very very interesting question, and really kind of highlights the complexity of transmission pathways in the hospital. There are certainly some people who are interested in the culturing of gowns and gloves of healthcare workers to actually sample nares, hands, and other kind of potential colonization sites of healthcare workers to kind of see their role in the transmission between rooms, but not something that we were able to answer here. And then lastly, is gender of the patient a factor? So that is something that we have planned to analyze, but did not make it into this report. But definitely, there's definitely a potential for gender to impact the potential contamination of surfaces. I don't know if it is that men are more likely to pick their nose and wipe surfaces, but I think that's I'll have to ask Michael David that sometime. If that may be why the near patient looks near patient environment looks awfully close to the nares. Any other questions? So for this particular study, we did not do water sampling, but that is, we presume that the sinks, the sink may splash into the sink drain and then may kind of spread Pseudomonas. And we thought that would be limited to the bathroom environment. So we presume that it's either the water or the kind of biofilms on kind of the drains and the sinks and the surfaces. But to tease that out, we could have done actual sampling of the water. We did not, just because of the kind of the complexity of adding that onto this study. But we are, interested in that is, there is a move to really separate patients from wastewater sites as there's kind of new hospitals are built, and so that may be something that it becomes less of an issue in the future. But if not, it certainly makes sense to tease out how much is the drain, how much is the actual water that is coming from the faucet. And that may change with time and temperature. So there may be kind of other factors that predict kind of how much Pseudomonas you have kind of coming out of your tap in that patient's room that could impact how surfaces in patients are contaminated. So not something we answered, but something hopefully we can add to a future study. Thank you for having me. Do I need to do anything to shut this down?